Bismillahirrahmanirrahim. Assalamu alaikum everyone. Welcome to Stream of Knowledge. Today we will discuss about the structure of bacteria. Bacteria contain essential components or essential structure that is present in all bacteria, while some species of bacteria also contain non-essential components. What are the essential components? These are cell wall, cytoplasmic membrane, intracytoplasmic structures that include nuclear apparatus or chromosome and ribosomes, while the mesosomes is also present as the essential component. And teichoic acid is also considered as a essential structure. Non-essential structures or non-essential components of the bacteria are the structure outside the cell wall, which are capsule, flagella, fimbriae, which is also called as pili, incluent granules, and other non-essential components are plasmid. Plasmid is present inside the cell wall. Now we will discuss essential and non-essential components in detail. As you can see in the picture, this is the cell wall. The cell wall is made up of peptidoglycan. Remember, peptidoglycan is present in both gram-positive bacteria and gram-negative bacteria, while this peptidoglycan or murein layer is thick in gram-positive while thin in gram-negative. Peptidoglycan is made up of peptides and sugar or glycan. That is why it's called peptidoglycan. What is the function of this peptidoglycan? Peptidoglycan maintain the shape of bacteria. It provides the rigid support so that the bacteria maintain its shape. It also protect against the osmotic pressure so that bacteria can survive in resistant environment. And it is the one of the major site of action of many antibiotics, for example penicillin and cephalosporins. While outside this peptidoglycan, there is also an outer membrane. It is only present in gram-negative bacteria, and this is lipopolysaccharide. Both these components are the part of cell wall. While in gram-positive, only peptidoglycan is the cell wall, while in gram-negative, both peptidoglycan and lipopolysaccharide is made up of can make cell wall. This outer membrane or lipopolysaccharide, which is only present in gram-negative, it acts as an endotoxin. Second essential component is plasma membrane, and this is the plasma membrane. Just inside the peptidoglycan, this plasma membrane, which is essentially same, which is similarly to the eukaryotic plasma membrane, but the difference is that eukaryotic plasma membrane contains sterols, while this is as the bacteria is the prokaryote, so it does not contain sterol in its cell membrane layer. This plasma membrane is the lipoprotein bilayer but without sterols, and this is the site of oxidative and transport of enzyme. In bacteria, there is an invagination of plasma membrane which is called mesosomes is also present. This mesosome is an essential component of bacteria. What is the function of this mesosome? This mesosome increases the surface area of the bacteria. Moreover, it participates in cell division and secretions. Fourth essential component is ribosomes. As we know, bacteria is a prokaryote, and the major difference between the eukaryotal and prokaryote ribosomes is that eukaryote ribosome is 80s, while prokaryote ribosome is 70s, made up of 50s and 30s subunits. And the function of the ribosome is the protein synthesis. Fifth essential component is the nucleus, and it contains all the essential information and contain DNA. The DNA of bacteria is single circular molecule. Remember, between the plasma membrane and the outer membrane, there is a periplasmic space. This periplasmic space is contain many hydrolytic enzymes, for example, beta-lactamases. This was all about the essential components. Let's move to the non-essential components. Non-essential components, or first we will discuss about those non-essential components that are outside the cell wall. For example, this capsule is made up of polysaccharide and protect against phagocytosis. Another structure outside the cell wall is the pili, or it's also called fimbriae, which is made up of pillin protein, and it have two functions. First is attachment to the cell surfaces, and second is conjugation. Conjugation is the transfer of DNA between two bacteria sexually. Another known essential component outside the cell wall is the flagella. Flagella is made up of flagellin protein, and it helps in motility. Now, the component inside that are non-essential are the granules. These granules, the cytoplasm contain several different type of granules that serve as the storage site for nutrients. For example, glycogen, lipids, and polysaccharides. Another non-essential component is the plasmid. Plasmid is also a DNA and contain a variety of genes for antibiotic resistance and toxins. Some bacteria also contain glycocalyx and and spores. That was all about the structure of DNA. Thank you for watching. Do subscribe, like, comment, and share. Stay tuned for the next parts. Thank you.